Hello, in this video we will discuss about the inositol triphosphate or Ip3 pathway, and we will also understand the primary messenger and second messenger of the cyclic AMP pathway for the glycogenolysis. So in this way, first of all, we should understand about the introduction of the first messenger. So the first messenger is the extracellular, basically is the extracellular ligands, can be epinephrine or other type of the hormone, which that is bind with the G-coupled protein receptor, GPCR. And after the binding to cascade of the second messenger, and the second messenger can be different. So here we will just understand about the Ip3 pathway as well as the cyclic AMP. So the first messenger is extracellular ligand, can be epinephrine, and it stimulates the second messenger. Second messenger can be intracellular molecules, Ip3, diacylglycerol, or calcium. This is the second messenger response intracellularly. So in this way, first of all, we will understand about the Ip2 phosphoinositol diphosphate. Basically, is the Ip2. In this way, you can understand as well. So the inositol diphosphate are present, and the phosphoinositol that is the basically Ip2. And in this way, here is the hydrophobic zone and hydrophilic zone. Hydrophobic zone is made up of glycerol, while on the other hand, the hydrophilic zone is the inositol phosphate. So after this cleavage formation to produce diacylglycerol as well as the Ip3. Ip3 is look like the hexagonal structure which contain a 3 phosphate group, carbon number 1, carbon number 4 and carbon number 6, carbon number 5 contain a phosphate group. So in this way, it is the Ip3 and DAG will be dissociation from the PIP2, and this mechanism we will understand about in the later on in this video. So first of all, we should understand about the Ip2 and Ip3. That is the mechanism for the secondary messenger from the primary messenger as a ligand. For example, ligand can be we will also understand about that. So here is the DAG formation and Ip3 formation. This is the secondary messenger will be produced, and this secondary messenger will play important role for the maintenance as well as the catabolism or metabolism with catabolism and anabolism in the cell intracellularly. So there are different response. So next, here we will discuss about the plasma membrane of the cell. So this plasma membrane contain many receptor, but here we will understand about the G protein coupled receptor. This G protein coupled receptor is intrinsic protein as well as the peripheral protein, the intracellular domain as well as the cytoplasmic domain. Cytoplasmic domain contain a G coupled protein receptor, while the trimeric protein are present in the intracellularly cytoplasmic protein, and which that is bind with the cytoplasm intracellularly. So in this way, the ligand can be glutamate, other signaling molecule, and ligand can be epinephrine, hydrophilic molecule, or some other. So for example, the growth hormone also. So in this way, the alpha monomer of the G-coupled protein receptor contain a GDP. After the activation by the GTP to remove GDP, and the effector will be formed. This effector will trigger the another adenylate cyclase enzyme after the activation by the GTP formation. So the alpha unit will be dissociate after the GTP formation. So the beta and gamma will be dissociate another way. So in this way, the effector can be adenylate cyclase and cyclic AMP. Cyclic AMP is the secondary messenger, and this cyclic AMP is the secondary messenger which that is lead to increase the sympathetic activity for the increase of the glucose, we will also understand how it's possible to increase the glucose from the muscle and liver. So in this way, the basically is the of a cyclic AMP will be increased in the cytoplasm. It is the second messenger will trigger the protein kinase A, and this triggering of the protein kinase A, the activation of the phosphorylase kinase P activation from the phosphorylase kinase by the using of ATP. And in this way, the phosphorylase kinase P is activated, and due to the phosphorylase inactive to become a phosphorylase P active. So in this way, the activation of the phosphorylase P activation for the glycogenolysis, and in this way, the glycogenolysis is basically here is you can see the phosphorylase inactive to become a active phosphorylase P. And in this way, the glycogen will be dissociated in the form of simple sugar by the using of the phosphorylase P active. So in this way, you can see in this diagram here. Here is the glycogen. Glycogen is the animal starch which that is stored in the liver as well as the muscle for reserve of the glucose. And in this way, the glucose will not require too much water when converted into the glycogen. So that is why the osmolarity maintenance will be important in our body. So that is why the glycogen formation, and this glycogen will convert by the phosphorylase P active to form a glucose 1-phosphate, and this glucose 1-phosphate is basically after the dephosphorylation to become a glucose. So in this way, here is the inactive glycogen synthase will be occur. In this way, the glycogen synthase inactive due to the glycogen synthase active to protein kinase A, which that is lead to inactive by the using of ATP. And in this way, the cyclic AMP is also occur in this way. And in this way, the glycogen synthase enzyme will be inactive. It means the negative response of the glycogenesis, while the glyconeogenesis will be occur. For the increase of the glucose during the sympathetic nervous system activation, and the sympathetic activity will be increased, and the blood sugar level in our body will be increased. So in this way, that is the cyclic AMP will be do like this. While on the other hand, the protein kinase is also activated by the cyclic AMP, and this protein kinase A is bound with a transcriptional factor to transcription of the DNA into the messenger RNA, and in this way that go into the nucleus, and the messenger RNA will be go into the through a nuclear pore to the cytoplasm, and translation will be occur. In this way, the translation can be cell growth, proliferation, cell survival, and migration, and metastasis. In this way, the excess of the protein kinase activity will lead to increase the cancerous cell due to the rapid growth and the maintenance and cell survival. In this way, the apoptosis will be inhibit, while the cell growth and cell proliferation will be promote. So on this mechanism, we will discuss also the other things you can understand as well. So let's begin to understand the two monomeric protein, G-couple protein, which that is G-couple protein will dissociate to dimeric, and this dimeric will here is the phosphoinositol diphosphate, which that is associated Ip2. This Ip2 is basically activated by the PLC and ion channel RPI3K, which that is the different pathway. So here we will understand just a Ip2 to Ip3 formation. So in this way, the phospholipase C will be activated and dissociate from DAG, and the IP3 will be formed. In this way, the IP3 will be formed, and this IP3 will lead to basically here is the protein kinase. We will understand later, but first of all, we should understand about the IP3. So this IP3 is the second messenger also. This second messenger is basically trigger the IP3 calcium channel in the rough endoplasmic reticulum or endoplasmic reticulum, and that is surrounded by the nucleus, and this is the basically the calcium will move out through a stimulation of the IP3. and the stimulation of the Ip3 is basically trigger the efflux of the calcium, and in this way the increasing of the cytoplasmic calcium level, and the cytoplasmic calcium level will trigger the protein kinase C inactive to form activate by the diacylglycerol, and this diacylglycerol will be activated by the phosphorylation of the P protein kinase C to active. And there are a lot of protein kinase C, for example the protein kinase D active and protein kinase C active, and there are many protein kinase C, alpha, epsilon, beta, and protein kinase C, D, 3, etc., and protein kinase D2. In this way, this protein kinase activation to lead insulin sensitivity will be decreased and ketogenesis will be decreased. In this way, the growth will be increased and gluconeogenesis will be increased due to the increase of the glucose level in the blood. So that is why it is the sympathetic activity, and cholesterol metabolism will be increased, lipogenesis will be increased, and glucose uptake will be also increased, and inflammation will be increased. Why? Because the increasing of the glucose and other molecules will lead to inflammation and the myoblast differentiation and the thermogenesis. On the other hand, the inflammation is also produce the chemokine from the cell of the lymphocyte, and insulin increase and apoptosis is also increased by the different manner. On this way, the other mechanism of the DAG, diacylglycerol. This diacylglycerol is here, which that is also converted into the monoacylglycerol. So in this way, here is the lipid droplets. This lipid droplet is contain a diacylglycerol is the same mechanism, and this convert into the monoacylglycerol, and the in the presence of the adipose triglyceride lipase enzyme, which that is convert the triacylglycerol into the diacylglycerol in the cytoplasm, in the in the membrane of the lipid droplet, and the DGAT is the hormone which, enzyme which that is transfer the DAG into the TAG or triacylglycerol from the diacylglycerol. And on the other hand, the hormone sensitive lipase will be activated, so increasing the monoacylglycerol also in the blood, as well as the hormone sensitive lipase will be increased, and in this way the diacylglycerol will be activate the protein kinase C by the lipid droplets. In this way you can understand as well.